Hello everyone, welcome to the online course of General Pathology. I'm Runhua Lin from Pathology Department, Shantou University Medical College. In this new chapter, we will talk about how tissues in the body react after injury. In other words, what are the measures the tissues can take for repair following injury? What are the differences between them? Does every tissue in the body use the same or different ways to repair? The leg of a 5-year-old boy was burned by the sodium hydroxide. This picture shows the appearance of the skin in his leg. We can see obvious scarring. Then, let's try to think, how can we explain the changes that took place? After this class, students should be able to define regeneration and contrast regenerative capacity of different cells or tissues. According to its definition, tissue repair refers to the restoration of tissue architecture and function after injury. But how do the body tissues achieve this? Basically, there are two types of repair. One is regeneration, and the other is fibrous repair. As shown in this diagram, when tissues with strong proliferated capacity suffer minor or superficial injury, they are able to replace the damaged components and essentially return to the original state. This process is called regeneration. But sometimes, tissues are badly traumatized, as shown on the right side in this diagram. The supporting structures of this tissue are severely damaged, or sometimes the injury is not serious, but the tissues have no or limited capacity to regenerate. Under such circumstances, repair can only occur by deposition of connective tissue. This process is called fibrous repair or scar formation. We should first understand the concept of regeneration. Regeneration refers to the replacement of lost cells by division and proliferation of adjacent healthy cells. However, tissue regeneration can be complete or incomplete. In the former case, the structure and function of the injured tissue will be completely restored. So, the regenerated tissue is the same as the lost one. Examples of physiologic regeneration are the continual replacement of epidermal cells in the skin and epithelial cells lining the gastrointestinal tract. However, in pathologic situations, complete restoration is also possible in tissues with good regenerative capacity. An example is the repair of the minor skin wound. Under what conditions the tissues would completely regenerate following injury? This slide lists some conditions required for complete regeneration. Firstly, the necrotic parenchymal cells have a strong ability to regenerate. As already mentioned, skin epidermal cells and gut epithelial cell lining are such examples. And secondly, the injury is minor or the supporting stromal framework of the injured tissue is well preserved. Actually, we often see incomplete regeneration in pathologic situations. During this process, the defect becomes filled by ingrowth of a specialized vascular connective tissue, called granulation tissue. The granulation tissue produces collagen to gradually form a fibrous tissue or scar tissue. This incomplete regeneration will lead to a loss in the structure and function of the injured tissue. Despite the loss of parenchymal cells and some architectural distortion caused by the fibrous scar, structural integrity is re-established. The regeneration of cells involves cell division. The key process in cell regeneration are DNA replication and mitosis. The sequence of events controlling this process is known as cell cycle. Some cells are continuously dividing. Most cells in the body are at the G0 stage of the cell cycle. These cells are quiescent and are not engaged in the replication or division process. Upon stimulation by growth factors, they may advance from G0 into G1 phase. However, the most specialized cells have little or no ability to divide. So, based on their regenerated capacity, we can divide body cells into three groups: labile cells, stable cells, and permanent cells. Labile cells normally undergo a continuous process of division and replacement, so we call them continuously dividing cells. If these cells are lost, they have a good chance of restoration by complete regeneration. Hematopoietic cells in the bone marrow and the majority of cowling epithelial cells belong to the labile type, such as the stratified squamous surface of the skin, oral cavity, vagina, and cervix. The lining epithelium of respiratory, gastrointestinal, and urinary tracts, and mesothelial cells. Stable cells have minimal replicating activity in their normal state, but they are capable of proliferation in response to injury or tissue loss. Therefore, we call them stable or quiescent cells. These cells constitute the parenchyma of solid organs, such as liver, kidney, and thyroid. They also include endothelial cells, fibroblasts, and smooth muscle cells. Permanent cells are considered to be terminally differentiated and have no proliferated capacity. So, we also call them non-dividing cells. If these very specialized cells are lost, they cannot be replaced. Examples of permanent cells are neurons, cardiac muscle cells, and skeletal muscle cells. This explains why injuries to the brain or heart is usually irreversible and finally leads to scar formation. But please note that peripheral nerve retains some capacity for regeneration following injury. Now, let's have a quick review of this section. Regeneration is the replacement of lost cells by division and proliferation of adjacent healthy cells to restore injured tissue. Based on the regenerated capacity, all tissue cells can be divided into labile cells, stable cells, and permanent cells. If tissues with strong regenerative capacity are only slightly injured, then repair is accomplished by complete regeneration. Otherwise, the repair would be incomplete with scar formation, like our 5-year-old boy with the burn. Here are the references, and see you next time.